Histology of cerebellum. This picture shows the general gross anatomy of the cerebellum. So as you can remember, cerebellum is located in the posterior cranial fossa, just beneath the tentorium cerebelli. And anteriorly it is related to the brainstem, mainly the pons and medulla. The pons and medulla, the brainstem is separated from the cerebellum by the cavity of fourth ventricle, and it is connected to brainstem by the three peduncles, namely the superior, middle and inferior cerebellar peduncles. Hope you can remember the gross anatomy of cerebellum and relations of cerebellum. Now generally before going to the proper anatomy of cerebellum, proper histology of cerebellum, let's see some general points. In sections of the brain and spinal cord, certain regions appear grayish, and these regions are known as the gray matter. And what this contains? They contain cell bodies of neurons and the dendrites and axons starting or ending on the cell bodies. And remember, most of the fibers are unmyelinated. Certain regions appears whitish. This forms the white matter. They are made predominantly of myelinated fibers. And what's the reason for the white color? They are because of reflection of light by the myelin. And another point is, arrangements of gray and white matter differ in different parts of the brain and spinal cord. For example, in spinal cord and brainstem, white matter will be the outer region, superficial region, and gray matter will be the deep region, inner one. Like this. Hope you all know this diagram. Outer white matter and inner gray matter. In cerebrum and cerebellum, gray matter will be outer and white matter will be the deeper one. So this is a section of the cerebrum, frontal lobe. So here you can see the gray matter is outside and white matter is the deeper one. Now we can go to the proper histology of cerebellum. The internal organization, the cerebellum, as we told earlier, outer one will be the gray matter, inner one will be the white matter. This is a hemisection of the cerebellum. You can see the gray matter and white matter over here very clearly. And you have studied the lobes and lobules of the cerebellum in the gross anatomy. They are further divided into folia or leaflets. Singular is folium, and each one of these are examples of folium. This is the superior view of cerebellum, and here also you can see these are the multiple lobes and lobules. Within the lobes and lobules you can see the multiple folia separated by the transverse fissures. And one exception is there. The gray matter is not only located on the superficial part, it also located on the deep part, that is within the white matter of the cerebellum, and these forms the deep cerebellar nuclei. Hope you all remember the 4 deep cerebellar nuclei in gross anatomy. The white matter fibers. Here you can see the term, that is arbor vitae cerebelli. As you can see, the white matter fibers of the cerebellum, they show characteristic branching pattern and they extend towards the cortical surface in a specific manner. This particular arrangement, that is known as arbor vitae cerebelli. And this white matter is consisting of afferent and efferent fibers traveling to and from the cerebellar cortex. Now we can see further into the cerebellar cortex and histology. So this is the general idea of the structure of cerebellar cortex. So in this picture, grossly you can see some white region, that is the white matter of the cerebellum, and superficial to that these are the molecular layer of the cortex of the cerebellum, and this is the surface lined by the pia mater. So how can we divide the cortex, this much region? It is divided mainly into 3 layers, and the layer which divides them is the Purkinje cell layer, which is the middle layer, which consists of a mono layer of large neurons, a single layer of large neurons. So the layer which is lying outside to the Purkinje cell layer, that is known as the molecular layer, and which is present deeper to the Purkinje cell layer, that is the granular layer, and here you have the white matter of the cerebellum. Let's see about each layer one by one. First one, molecular layer. As you can see, it is lying outside to the Purkinje cell layer. It mainly consists of neurons and fibers. There are mainly two types of neurons are present. One is the stellate cell, which you can see. As you can see, it's lying on the outer part of the molecular layer of cerebellum. Another one is the basket cell, which is lying on the deeper part of the molecular layer of the cerebellum. In addition to these neurons, they contain certain fibers also. One of them, major one, will be the, not major one of them will be the parallel fibers. The parallel fibers are originally the axons of the granule cells, which is located in the granular layer of cerebellum. They traverse through the Purkinje cell layer, then there is the molecular cell layer, then they divide into a T-shaped formation and forms the parallel fibers. Next type of fiber present in the molecular layer is dendritic tree of the Purkinje cell. This is one Purkinje cell, and you can see the arborescent dendritic tree of the one Purkinje cell. And the important point to note is they are arborescent in one plane only, in the sagittal plane mainly. Next type of fibers present in this layer is the dendritic tree of the Golgi cells. This is an example from one Golgi cell, and these are the dendritic tree of Golgi cells, and they are arranged in all the planes, not only in sagittal plane but in a transverse plane also. The next layer is the Purkinje cell layer. The Purkinje cell layer, as I told earlier, it's a single layer consisting of cell bodies of the flask-shaped cell bodies of the Purkinje cells. Now, from the outer part of the cell body, dendrites will be arborescing in the molecular layer. From the inner part of the cell body, axons will be passing into the granular layer, then it will reach the white matter of the cerebellum, then it will go towards the deep cerebellar nuclei and vestibular nuclei. So this is an example of one Purkinje cell. As you can see, this was the basket cell we have saw earlier, and the axon of the basket cell form a basket around the body of the Purkinje cell, and they are connected to the initial segment of the axon of the Purkinje cell. So this is a picture depicting the orientation of Purkinje cell in our body. So if you keep our right hand like this, these fingers will be representing the dendrites, and this portion will be representing cell body, and forearm region will be representing the axon, all of them lying in the sagittal plane. So this is a HND section. Here you can see the Purkinje cell bodies, and here you can see the molecular layer. Here you can see the granular layer. As you can see, the Purkinje cell dendrites are arboresced into the molecular layer. Here you can see the granular layer of the cerebellar cortex. Similar to the molecular layer, here also you have two types of neurons, that is the granule cell and the Golgi neuron. This is the granule cell, is the yellow colored one, and this is the Golgi neuron. And the dendrites of the granule cell and axons of the Golgi neuron are located within the granular layer itself. Other than that, you have two types of fibers, main type of fibers, that is the climbing fiber and mossy fiber are present within the granular layer. Actually, they are passing from the molecular white matter of cerebellum, and they are passing towards the cortical layers. So they are also present within the granular layer of cerebellum. Then you have the axon of the Purkinje cell, which is passing towards the white matter of the cerebellum, deep cerebellar nuclei of the cerebellum, through the granular cell layer. So that's also present within the granular cell layer. Here you can see some structure. The mossy fibers, if you trace the mossy, it will form some structure over here, that is the glomeruli of the cerebellum. And what is the glomeruli? So here you can see the mossy fiber terminal, that is giving rise to a rosette, and here you can see the granule cell dendrite, and here you can see the Golgi cell axon. Together they form the glomeruli of the cerebellum. The granule cells within the glomeruli, they receive the impulses from the Mosey fibers, and the dendrites of granule cells and axons of Golgi cells, they synapse with the terminals of Mosey fibers within the glomerulus of the cerebellum. Now major afferent fibers to the cerebellum, they comprises of climbing fiber and Mosey fiber. We have seen them already. Mosey fiber comprises of all the afferents to the cerebellum except the fibers from the inferior ovary nucleus. The fibers from the inferior ovary nucleus forms the climbing fiber. Then both are excitatory. Efferent fibers of the cerebellum, axons of the Purkinje cells are the main efferent fibers of the cerebellar cortex. As you can see, they are inhibitory to the cerebellar nuclei. I told you earlier, these fibers from the cerebellar cortex will be passing towards the deep cerebellar nuclei located within the white matter of the cerebellum. This is the histological diagram picture showing the cerebellar folia, and can you label? 1 is the molecular layer, 2 denotes the Purkinje cell layer, 3 denotes the granule cell layer, and 4 is the white matter of the cerebellum within a one folia. And this much portion of cerebellar cortex is enlarged to show how the Purkinje cells are arranged in a H&E section in a hyperview. So this is the Purkinje cell body. You can very well see that the dendrites are branching, arborescing within the molecular cell layer, which is very cell sparse layer. And deeper to that, you have very rich cell rich area, that is the granule cell layer. And within the granule cell layer, you can see some lessly stained areas. They are the glomerulus or cerebellar islands. This is how you have to draw the diagram. You have to show the 3 layers of the cerebellar cortex, and you are showing the one folia over here, cerebellar folia, and the white matter of the cerebellum is extending into the cerebellar folia. You have to show the Purkinje cell layer in an equally spaced single layer. Okay, thank you.